Welcome back, everyone. So we're going to finish off our Tools of the Trade Topic 1. So we've completed most of Topic 1 already. So we're going to finish off with microscopy. So in this final part, we're going to learn about light microscopes and how they're used in the field of biology, as well as describe cell theory. So as always, good idea to write these learning objectives down. And then by the end of this video, see if you can answer these two objectives. So what is cell theory? Well, first off, do you remember what a theory is? So a scientific theory is basically like a hypothesis on steroids. So it's a guess that has been, an educated guess that has been, it's broad in scope, and it has been proven over and over and over again with varied evidence. So in this case, cell theory states that all living things are made of cells. So far, we have never found a living thing that is not made of cells. So all living things are made of cells. So that's part one of cell theory. So everything that is alive, has ever been alive, has at least one cell in it, okay? And then the other part of it is that cells come from pre-existing cells. So we have never found a cell that just poofed out of nowhere, okay? We've never seen that happen. So it always came from a pre-existing cell, okay? So even you came from two pre-existing cells. You came from a sperm and an egg fertilized together to form a zygote, a single cell, which divided and divided and divided to become you. So cells come from pre-existing cells. So that is the essence of cell theory. So all living things are made of cells, and cells come from pre-existing cells. Okay? And then as far as cells itself, when we talk about living things, some living things are just made of single cells. So we can think of bacteria, some protists are like that. So some organisms just are one single cell, or some organisms are made of many cells. So think of any animal, so humans, cats, dogs, fish, all made of many cells. Plants, fungi, things like that are all made of many cells. And then we can also categorize cells into two groups. They can be simple cells, which we classify as prokaryotic. Those are cells that don't have a nucleus, and we'll go into more detail about that later, versus complex cells that do contain a nucleus. So those are called eukaryotic. And so when we're talking about like classification, plants and animals and fungi and protists, they all have eukaryotic cells. They all have these complex cells that have nuclei. So if I took a look at your cells under a microscope, they would each have nuclei. Okay, if I took a look at plant cells under a microscope, they would have nuclei. Okay, whereas if I took a look at bacterial cells under a microscope, they do not have nuclei, so they're more simple, so they're prokaryotic. So we can kind of classify cells based on whether they have a nucleus or not. So prokaryotic cells don't have a nucleus, they're simple like bacteria. And then eukaryotic cells do have a nucleus, they're more complex like plants and animal cells. Okay. So now let's get into microscopes. So most likely when we encounter a cell, we cannot see it with the naked eye. With some exceptions. So for example, a chicken egg is one single cell, and you can certainly see that without a microscope. Probably, one of the larger cells that you can see, an ostrich egg. Probably the largest cell on Earth, I would say, would be a neuron in a blue whale that can be 6 feet long. So there are some very large cells out there. But as far as human bodies, we cannot see any of our cells with the naked eye. So we would need a microscope to examine our cells. So most cells cannot be seen without a microscope. So a microscope is just an instrument that we can use that will magnify an object for us so that we can see it better. And what a microscope does is it basically it has two lenses inside of it, and so it can magnify objects. So the ones that we use at school, it's a light microscope, so it basically bounces light off of these lenses to help magnify those objects. Now when we use a microscope, it actually inverts the image or the object. So for example, if I'm looking at, if I put a slide of a letter E in the microscope right side up, when I'm looking at it under the microscope, I will see it upside down. Okay, so it's inverted. And that's because of the lenses. So I'm basically seeing a reflection off of it. Okay. So there's actually two types of microscopes out there. So one of them is something that you've probably used if you've ever taken any type of lab before, which would be a light microscope. So that's the one that's more familiar with you. The other type is an electron microscope. So we're going to talk about both types. So light microscope and electron microscope. So we'll start with the light microscope. So the light microscope, again, is the typical one that you would have at school. So in this case, it's basically we're using light that passes through a lens so that we can see the object. Now these microscopes, they usually can have an objective up to 400 times magnification. So if we're looking at something under a microscope, we can magnify it, usually up to 400 times, which is pretty good. I mean, we could see things like our blood cells under a microscope. We can magnify it, 400 times. That's pretty cool. Another advantage of light microscopes is we can see living things. So if you've ever done this in school before, you could take like pond water and you can put it under a microscope and you can see all the little critters and they're still alive, moving around under the light microscopes. And so, it could be magnified, 10 times, it could be magnified, up to 400 times. So it can be very useful in a classroom setting. So when we're looking through a light microscope, we can not only use the magnification, so that's basically magnifying, seeing things closer. And then light microscopes also have resolving power. So resolving power is the ability to distinguish two objects as separate. Okay, so there's to a certain degree. So if our light microscopes can't go too far up in magnification, because then we would lose this resolving power. So we, eventually we won't be able to distinguish two objects as separate. Okay. Some of our light microscopes, like at our ones on campus for our, actually I think the ones that we use in our 101 course, some of them do have 100 times objective lens, which would allow for 1,000 times magnification. If those are present on our microscopes, students can use them, but they have to use them with oil. So that's called an oil immersion lens, and that can again allow for us to see things up to 1,000 times. But again, that's usually doesn't work too well in a classroom setting because it's very, very likely that a student can use it improperly. So most of the time we just use up to 400 times magnification. Okay. So the light microscope, definitely fun. Unfortunately, we don't get to use these in the online setting, but you will get to see some images in our workbook. Okay. Now the electron microscope is different. So I've actually never used one. They're very, very expensive. When I was doing my master's degree at Cal State Fullerton, they actually had one. It was like deep in the basement somewhere, and I wasn't even allowed to go in there. You had to have like special clearance. I think they are like $1 million. So it's basically, you have to have special clearance to even go near it, because if you break it, then you're basically screwed. I wasn't allowed anywhere near it because I had no reason to use it. I studied birds, so there's no reason for me to use it. But it uses a beam of electrons instead of light. And because it uses a beam of electrons, you can't use live specimens, because it would kill it, kill the live specimens. So since I was looking at birds, I was studying bird song, I'm not going to kill any birds with an electron microscope. So I was nowhere near any of this equipment, but it allows for extremely high magnification, so you get really good detail. So if you see here, here's some examples of some images from an electron microscope, and look at the size here. This is 25, you see this weird U, that's micrometers. So if you looked at the previous video, what's micro? Micro was 10 to the minus 6. Okay, so that's 0.00000. There's 6 zeros there after that decimal point. So that's really, really small. So that's, you're getting very, very magnified here. So that means that we can see inside of the cell. And we can see like little organelles, okay? And sometimes we can even see like molecules inside the organelles, like we can see proteins. So this is 1 micrometer. So I mean, you can get into like little proteins and DNA and stuff like that. So this is highly magnified, okay? So electron microscopes, very cool. You can get highly magnified, but you cannot use live specimens. And they're very expensive, and we do not have one at Clackamas. I don't know if there is one in the state of Oregon. Usually what happens is there might be one somewhere that universities share. So like I know that Cal State Fullerton had one, and it shared it with like multiple universities in the area. Okay, so I don't know if there's one in the state of Oregon. All right, so here's a review question for you. Light microscopes used in student labs magnify specimens up to, is it A, 4,000 times, B, 100 times, C, 400 times, or D, 40 times? If you chose 400 times, I would agree with you. So they typically go up to 40 times, or 400 times, 400 times. Okay, so we usually have that 400 times magnification. Occasionally you'll see the ones that can go up to 1,000 times, but that requires oil, so not always available. Okay, so C is the correct answer. Okay, so that is the end of our microscopy topic. So by now you should be able to answer these 2 questions: explain how light microscopes are used in biology, and describe cell theory. This also ends our topic 1, tools of the trade. So we have finished our textbook reading. If you haven't had a chance to look through the textbook, make sure that you do so. We've covered all the questions, so you can just go through and answer those questions so that you get those points for the grade book. Okay? If you have any questions, feel free to reach out. Otherwise, go ahead and start working on your workbook if you haven't done so already.